and welcome to part 3. So we're talking about the heterochrony. This is the timing gene. So how long do the bones grow in the neck? So in humans, like, okay, the genes say start growing now, and they grow, and then it says stop. And then if that gene is delayed, you'll get a longer cervical vertebrae. Giraffes have the same number of cervical vertebrae as humans do. Those bones are just longer. They didn't get, the growth didn't get turned off. Same thing with the stripes of a zebra here. You get the gravy zebra, which is skinnier stripes. That's simply because the timing of that formation of their skin stops sooner. So timing is very important in the pattern formation and morphogenesis. This is another example of how nearby cells actually do manage a lot of this process. So if you take an animal that has small eyes and you put those genes in the DNA of a large cell animal, the large-eyed animal is still going to grow large eyes, even though it has the small-eyed gene in it. That's because the neighboring cells actually do a lot of the controlling of it, of that gene, or of that formation of the eye. Genes on the Y chromosome can influence morphogenesis. Remember, the Y chromosome is found in male but not female. The gene SRY is found on the Y chromosome. It stands for the sex-determining region on the Y chromosome. And that gene will put off factors, gene regulation factors, that will turn the sex organs into testes, the gonads into testes. And then the testes will set forth a number of other things that will make the things like the epididymis and the vas deferens that are all part of a male reproductive system. If there is no SRY gene, then the gonads will develop as ovaries, and instead of developing the epididymis or the vas deferens, they'll develop fallopian tubes and parts of the vagina and things like that. So, and female reproductive system. And it's all determined kind of by this SRY gene that kicks off the cascading effect. And this is showing, more of that. SRY, the genes on the Y chromosome that can develop gonads one way or another. The development effect of testosterone on development is also rather big, and estrogen is also rather big. If you have the SRY gene, you're going to make the gonads into testes, and then that'll set forth a series making the Leydig cells, which will make testosterone, which will do a lot of other things. If you don't have testes, then you'll have no Leydig cells, you'll have no testosterone, and you won't get all of the gene expression that comes with the testosterone. That's definitely a gene expression factor if you're a female. But if you have the testosterone, you'll have it. Interesting, cloning. People didn't think that if you had a differentiated adult cell, which means that it's already been differentiated in some way from the totipotent or pluripotent cell, that you couldn't make a clone from it. But they actually proved that you can, even though the cloned organism has lots of problems, like shorter lifespan, premature arthritis, obesity, liver failure, things like that. But this slide kind of talks about how they did that in the making of Dolly, the first genetically identical organism. Watch these videos, and we will see you soon, hopefully. Love you. Find some time to pray and connect and stay healthy. Definitely miss you a whole bunch. That's it, though. See ya. Bye.